The synthesis of DNA is catalyzed by an enzyme which is called DNA polymerase. Rather, you can say DNA polymerase is the most important enzyme during the process of DNA replication. This enzyme has a single active site, and it uses this single active site to catalyze the addition of any of the 4 deoxynucleoside triphosphates. You know, there are 4 types of deoxynucleoside triphosphates, and any one of these 4 can come to join with the existing template. But this polymerase, this identifies the correct DNTP or deoxyribonucleoside triphosphate to be joined with the template. So it has only single active site, and with this single active site it identifies all these 4 types of DNTPs. DNA polymerase monitors the ability of the incoming nucleotide to form an AT or GC base pair. So this enzyme is monitoring which nucleotide is coming and what type of base pairing is to be taken place. For example, A will only be a base pair with T, and G will only base pair with C. So this is correct base pairing, and this is monitored by this DNA polymerase, and it doesn't detect the exact nucleotide that enters the active site. So any of the 4 DNTPs, they can come to the active site, but only the right DNTP will be added to the existing chain of the DNA. Only when a correct nucleotide comes, only and only then the 3' hydroxyl of the primer and the alpha phosphoryl group of the nucleotide, they align in the optimum position so that the catalysis can take place. So this is the steric position, this is the position in space of the 3' hydroxyl and the alpha phosphate group. So they only become in front of each other when a correct nucleotide will be present. For example, here you can see this is the addition of new nucleotide. For example, here in this diagram you can see this is the template and this is primer. So in this template, this adenine is present, and you know adenine will only base pair with thymine. So here the incoming DNTP is thymidine triphosphate, DTTP. So when it will base pair, here you can see the orientation of 3' hydroxyl group and the alpha phosphate. They become in front of each other. So only in this way this hydroxyl can attack the alpha phosphate of the incoming nucleotide. While in this case, in this figure, you can see here it is adenine, and the incoming nucleotide also contains adenine, but here this OH that doesn't come in alignment with the alpha phosphate, and it cannot attack the alpha phosphate, and the addition of new nucleotide cannot take place. Incorrect base pairing leads to dramatically lower rate of nucleotide addition. If incorrect nucleotide comes, the addition of this incorrect nucleotide is very rare, or very less chances of addition, because the catalytically unfavorable alignment of these substrates will take place, and the catalysis will not proceed. Only and only this catalysis will proceed when correct alignment will take place, and correct alignment will only take place when the correct nucleotide will come to join with the existing template. DNA polymerase shows an impressive ability to distinguish between ribonucleoside triphosphates and deoxyribonucleoside triphosphates. As you know, these RNTPs and DNTPs, they have a very slight difference, and the difference of only one O is present between these RNTPs and DNTPs. So there are chances that RNTP can base pair with the template instead of DNTP, but DNA polymerase, it has very good ability to distinguish between RNTPs and DNTPs. Although RNTPs are present at approximately 10 folds higher concentration in the cell, yet their incorporation rate is 1000 folds lower than the DNTPs, and that is due to the ability of DNA polymerase to distinguish between RNTPs and DNTPs. This discrimination is mediated by the steric exclusion of RNTPs from the active site of DNA polymerase. This is similar as you have seen in case of wrong DNTP, which has come to join to the template, but the catalysis doesn't take place due to ill alignment of 3' hydroxyl and beta phosphate group. So in the DNA polymerase, the nucleotide binding pocket cannot accommodate a 2' hydroxyl group on the incoming nucleotide. See, this distinction between RNTP and DNTP, that is due to this reason, that is the nucleotide binding pocket of DNA polymerase that cannot accommodate a molecule which contain 2' hydroxyl group, and 2' hydroxyl group is present in RNTPs, while in case of DNTPs at the 2' of deoxyribose there is only hydrogen present. There is one less oxygen at 2'. This space is occupied in this pocket. This space is occupied by 2 amino acids that make van der Waals contacts with the deoxyribose ring. These amino acids are called discriminator amino acids. So here in this you can see this is the discriminator amino acid, and this is correct DNTP. When RNTP with this hydroxyl group will come to join with the template, the correct alignment will not take place, and catalysis or the addition of this nucleotide will not take place.